Hello, my name is Ben Thompson. I'm a PhD student at the University of Michigan in Dr. Satin's lab. And I'm just making a quick little video sort of giving a virtual tour of our lab for people who are potentially interested in joining or just want to know more about what instruments we use and the kind of work that we do. Our lab is located on the 5th floor of the Brem Tower, which is part of the Kellogg Eye Center over here, which is just sort of its own little island away from the main medical campus, which you can see right up the hill over there, that being the cancer center and the main hospital. Because it's on the 5th floor, one thing that's nice is that we have a really great view. Some of these windows are sort of closed. Some recently built parking structures, so parking is never an issue, as well as this very large atrium that people use to eat and sort of relax during lunchtime or any other time during the day. So now entering into the actual lab space, it's a little loud just because of all the fridges and freezers, so apologies if you can't hear me too well. But the lab's sort of broken up into two different sections. You got this first hallway, which contains all the fridges, freezers people use to restore their reagents, as well as the cold room, which is right here. Nice and spacious, that's shared between all the labs, as well as a couple of our own rig rooms. So the first rig room, just going sequentially through, is a patch rig, which I use, as well as my office. And just closing the door because of the loud sound from the air conditioner. This is, like I said, our first rig room, sort of storing a bunch of equipment and little tools that we use to repair and fix up our equipment to make sure everything's running properly. Got my desk, a couple of recordings on the wall. And then over here is our first patching rig, sorry. So it's used as an inverted microscope by Olympus, and gravity perfusion through some tubes, through a regulator into our chamber, and we use to record membrane potential and currents using our little head stage that's currently disassembled as I'm currently fixing up some issues with it. We use Heka patch clamp EPC10 USB, an MP285 manipulator, as well as Patchmaster software on the computer. At the other end of the hallway, we have another rig room that's used for live cell imaging. So this is also using an inverted microscope, gravity perfusion with a Lambda XL lamp. And we often use this rig simply just to measure cytosolic calcium, although if you change out the filters, you could record other different fluorescent probes. Sort of a smaller room, really cozy. It gets really warm in here, which is really nice for the winters. And it's a little ways from our lab, but it's a great room to just solely focus on imaging. Now moving into the main lab space, you can see that we have an open lab environment where all the labs on the floor will work on beta cells or islets, measuring insulin secretion and usually studying some form of diabetes. So starting all the way at the end over there is Dr. Ling Key's lab, followed by some metabolomics core equipment that's left over. They're also upstairs. So really nearby, if you want to do any metabolomic studies. Got Dr. Scott Solomon-Poor's lab, and then the introduction of our lab. So our lab is made up of 3 different bays. The first one we're just using sort of to hold some equipment. The second one, and the main bay that we use, we have some equipment and stuff to do aisle isolations, which would be at this desk right here. Centrifuge for spinning down equipment. A plate reader that's shared by everyone on the floor, as well as one of our pullers to make glass electrodes for EFIS. On the second bay, this is mainly our bay that we use for wet lab research. So PCR, Western blots, and any sort of other molecular technique. And then at the end of all these bays, we have some desk space available with some computers. Some of us like to use our own laptops for research. And at the end of each of them is a nice big window, so lots of natural light that comes in, and a sort of a good view looking out. Right now it's middle of February, so we have a lot of snow on the ground. But in the summer, it actually looks really nice if all the leaves on the trees and stuff in bloom. So next to our lab is Dr. Peter Arvin's lab. That makes up this bay right here, as well as the room over there. So some shared equipment space is right here, which is our chemical room and our glass. So this is our glass cupboard, as well as a couple of hoods, scales, and such. Our chemicals that we use to make our glucose solutions and other various solutions that we need for perfusion. And then as we walk into our lab, we have a couple of rooms solely just for our lab, more equipment rooms. This first room right here is what we mainly use for live cell imaging and to do FRET. So it's just a computer that runs Metamorph software. And then we have our Polychrome V lamp, which is used to excite our microscope. This one's also an inverted Olympus with our DV2 beam splitter and quantum EM camera. Again, it's a nice sized room, heater on the bottom. And this room is mainly used just for live cell imaging, usually FRET using different probes like our PCAR probe, which measures intracellular FBP, D4ER, which measures ER calcium, mitopericam, which measures mitochondrial calcium, and other various probes that we have that we can express in beta cells. This third room is a larger room that has two different microscopes on it. Starting all the way at the end, this is our second patching rig. This one is an upright Olympus, but it also runs the same Heka patch clamp EPC10 and Patchmaster software. Basically similar setup, just in a different room. And over here is our cytosol calcium rig, our second one that's in the main lab, that was recently being upgraded with a new camera, an LED light source in the back that you can't really see, sorry. And also uses gravity perfusion in order to run glucose solutions over the cells in order to measure calcium oscillations. Last but not least, our last room for our lab is our tissue culture room. It has sort of your standard tissue culture equipment. We have a microscope here for picking cells and islets. Our refrigerator, which contains all of our reagents for doing islet isolations and other various experiments. We have our tissue culture hood. And then we have our incubator. So that's sort of a quick summary of our lab. So in total, we have 3 rigs, which can measure or do light cell imaging. We have 2 patching rigs, capable of measuring membrane potential and cell currents from islets and other cells. We have our tissue culture lab, a shared chemical lab, and a couple other spaces that I didn't show because they belong to other labs. But it has more equipment in there that we can use for Western blotting development and PCRs, Northern blots, whatever other kind of wet lab techniques we want to do. But all in all, it's a great space. There's a lot of big windows, natural light that comes in. Everyone is very friendly, and all the different labs collaborate with each other. I personally have been working in this lab for about 7, going on 8 years now. And I personally love it, love all the people that work here. All the labs are very open and friendly with each other. All the PIs are incredibly intelligent and smart. And it's a great place to work, in my opinion. So I hope you enjoyed. If you have any questions, feel free to email me, bmthomp@umich.edu, or you can contact Satin for more information. Thanks.